Okay, welcome everyone. Hi. Um, Sorry. So, good morning or good morning or good afternoon, depending on what time, where, where in the world you are joining us from. Thank you for joining again the session of the Public Journal Club. Apologies for the problem we had. Um, we have last this time of the time, but we will start now. Before we start, I like to today's paper discussion. I would like to tell you about this project. This public journal club is a paper discussion space created by grads and soon to be grad students for grad students, so and students in general. So we mainly focus on immunology related topics, but the main idea is that everyone contributes to, a different point of view depending on their background. We usually take around 45 minutes to an hour to discuss the paper. And after that, we have arranged a small questions and answer session with the first or last author of the publication. This time, we'll have Arturo Ciklinski, and we will discuss neutrophil extracellular traps kill bacteria. Thank you again for joining us and making this project grow. So I would also like to invite you all, if you are interested in proposing a paper for discussion, or even better, if you have published recently your work and you would like to join as an author, please write us in our social networks, in Twitter as @publicjournalc, and Facebook and Instagram Public Journal Club, and let us know. So please, before you start, please turn off your camera and microphone so we don't have problems with the network. And we remind you that this session is being transmitted on YouTube, for you to, if you want to join that way or want to see it afterwards. As I said before, today we will not discuss a recent paper, but a very important one for immunology. This paper was made by Arturo Ciklinski lab, and as I said, it's Neutrophil extracellular traps kill bacteria, a paper that was published in 2004 in Science magazine. And since then, it has been cited more than 4,000 times in different contexts. This paper reported for the first time the mechanism of neutrophil extracellular traps formation and its function in immunity. This paper was just the beginning of our understanding of processes in biology, and today you can find hundreds of papers and reviews discussing about NETs. Today's paper will be presented by Carlos Alfonso Gonzalez. He's a PhD student at the Max Planck Institute in Freiburg, and he is mainly interested in RNA biology. As I said, after a 45 minutes paper discussion, Arturo Ciklinski will join us for questions and answers. Thank you and welcome everyone.
You can hear me, right? Yeah. In the case of the authors, Arturo Ciklinski is the director of Max Planck Institute of Infection Biology in Berlin.
Sorry,
and
Carlos,
the first author.
Carlos, we couldn't hear the first things you said.
Can you hear me now?
Yeah, but we lost your first slide.
Okay. Yeah. So yeah, I'm gonna present you this, and thank you very much everyone for being here. And yeah, about the authors. So the authors who published this paper, the group leader was Arturo Ciklinski, which is the director of Max Planck Institute for Infection Biology in Berlin. and he will join us in 1 hour. And the first author of this paper is Volker Brinkmann, and he's now the director of Novartis Institute for Biomedical Research. It's interesting. It's important to mention that Arturo Zieliński is still working on neutrophils and nets, but additionally he have a line of research centered in Drosophila, using Drosophila melanogaster as a model, in which he studied histones in immunity, but also the role of histones during development. So as a background, we don't start with the classic background, so that normally people do in papers. What I'm going to show you is basically what was known in 2004 to put in context how important this discovery was. So at that point in science, in neutrophil science, it was only known that neutrophils would cover most, close to 60 to 70% of all mammals' blood cells. And it was also known that these cells were the primary responders to infection, and also that they have a very particular feature, polymorphonuclear characteristics. One thing that is very interesting about neutrophils is that neutrophils engulf bacteria to basically to clean out bacteria from infection. They clear bacteria by means of phagocytosis, which is a process in which the neutrophils engulf bacteria, and then this puts the bacteria in close proximity to the proteasome and promotes bacterial removal. This phagocytosis works by means of proteolytic enzymes, antimicrobial proteins, and ROS. And one characteristic very important that was known at that time is that neutrophils degranulate. And this observation that neutrophils degranulate was made before, saying that this degranulation would release some factors into extracellular media that would also exert antimicrobial response, like will help the antimicrobial response. So, and this also these discoveries were made by the Zieliński lab before. So this is what led to what led basically to the paper. So in summary, there was not much about, there was not a lot of things known about neutrophils. Mostly it was just that they phagocyte bacteria and that they just do it by means of granules and that they also secrete granules. But the first observation of the article, and this is what leads to the discovery, is that nets, after they are activated either by interleukin 8 or PMA or LPS, the shape of the neutrophils changes from a stereotypical spherical cell to a very flat and full of protrusion cells. Additionally, these cells, they observe that they start forming laminar structures like surrounding the cells. So they ask, well, like they study like closer what were these structures, and in the paper they mentioned that these structures were particularly hard to isolate and to visualize, but they manage. And basically what they see when they did electron microscopy is that these structures are with a diameter of 15 to 17 nanometers. And yeah, so basically it was the first observation. Upon activated neutrophils with EL8, PMA or LPS, you can start seeing these structures that have a like a net shape. So the next thing that they did is that the laboratory of Arturo Zieliński described first that neutrophils attack bacteria using neutrophil elastases. So they went to check if these nets were formed by neutrophil elastase, and indeed the expression of these structures was full of neutrophil elastases. Additionally, if they did also stainings for DNA, and they see that there's a perfect correlation, almost a perfect correlation between the neutrophil elastase and the DNA. Additionally, they also looked into if dsDNA would still have chromatin complexes like histones, and also they found that. So in summary, they found that there were some structures that were activated upon neutrophil activation, and that these structures would be made out of chromatin complex with neutrophil elastases. Additionally, they corroborated these results with correlation microscopy. And basically what they did is they look closer into to make sure that this was indeed the case using electron microscopy. And then they found that the neutrophil elastases and the chromatin form complexes. So in here in the arrows, you can see both the elastases and the histones, and you see how well they correlate with each other.
Before you continue,
so this
Carlos, does anyone wants to add something on the previous figures? If someone has a comment or a question, please feel free to write in the chat or open your mic and ask. Yeah, so if someone has any comments. One thing that I think it's important to mention is in the previous figure, the previous slide, when they show that there's DNA neutrophil elastases, but they show a complex of DNA and the two histone proteins. They are not showing just that there has histones, but there's like possibly full nucleosomes in the nets. So that's very interesting that there's still chromatin structure or chromatin related proteins attached to the DNA. So we can continue.
So yeah, so after these observations, the first thing that anyone would think is that this is just the result of leakage, right? That this phenotype that we're seeing is just the result of cell death. And the authors in the paper basically define why they believe that the net formation is a regulated process and not just some accidental mechanical response. So one of the things that they claim is that the nets, one of the things that they found is that even though that they found markers for some of the primary and secondary granules of neutrophils, they do not find cytoplasmatic markers, which means that this is not just any this is not an accidental burst, nuclear burst. Additionally, they also say that stimuli that prolong the life of neutrophils can also induce nets, meaning that you don't make nets and die immediately. And also, incubation of DNA intercalating dyes before neutrophil activation prevents net formation independent of apoptosis. And also, they found that they also claim, so these are all observations of things that were already known, but they just put these observations in the context of net formation. And additionally, and one of the major claims is that motile cells also make nets. So this is not necessarily that the fact that there's that the neutrophils are migrating, that they will start just releasing, like they lose intracellular contents. So based on this assumption, they try to find what's the function of nets and what kind of antimicrobial response the nets can do. So well, and here this is the end of, okay. So one of the things that they find, that the first claim is here about the net function itself, is that they found that nets degrade virulence factors, and this effect it's independent of gram-positive or negative bacteria, which is, I think, very interesting. So they found that both gram-negative and gram-positive bacteria were susceptible to nets. And they even tested this using different proteinase inhibitors, and they found that in both cases the virulence factors, this is IPA-V, in this case IPA-V is the virulence factor, and you see how the interaction of the net with these factors reduces its amount. Additionally, they also tested whether this process was paired with phagocytosis, because as I mentioned you at the beginning, the main described role at that point was that neutrophils do phagocytosis. And basically, they asked what happens if they inhibit phagocytosis using cytocolastin D, and they wanted to see, like, if they inhibit phagocytosis, they would still have the disarming of the viral or the virulence factors, and this is what they find. So they have a degradation of both virulence factors, even if they treat with cyclostolastin, which means that it's an inhibitor of phagocytosis. We see a reduction in both levels of virulence factors, while as a control of a membrane protein in bacteria, this one is not affected. So with this, they conclude that nets disarm a wide range of pathogens, but they also do it specifically targeting the virulent factors that bacteria release, not necessarily the main bacterial component. So there's some specificity of nets in disarming virulent factors.
Does anyone wants to comment on this?
Hi, can you hear me?
Yep.
Yeah. Oh, yeah. I wanted to ask you, like, do you actually think that, like, I don't know, but this seemed to me maybe a bit exaggerated statement, you know, based on them. Like you can see that indeed the membrane components of the bacteria that are not virulence factors seem not to be affected by the nets, but at the same time we are just looking at basically two examples. So do you think that there is like a further reason of why they could actually make this statement, or you think it was just you know, a bold guess done at that time?
I think this statement comes from a previous paper that also the lab published, in which, so I think so before it was known that neutrophils have one particularity, that once the bacteria enter to the neutrophil, it's very hard for the bacteria to escape, something that does not happen with macrophages or epithelial cells during phagocytosis. So one of the things that the Silchinsky lab found is that bacteria can escape this by releasing virulence factors. So the fact that the nets are able to reduce the amount of virulence factors in both negative and positive bacteria is that makes the claim kind of strong, that that nets are do not require that much of a specific response.
I think it is also based on the fact that the neutrophil elastase, which they have published before, the neutrophil elastase has like antimicrobial activity and can degrade these exact virulence factors. So now they are just testing if the neutrophil elastase in the nets is active, which I think it was very interesting, that it was not that the proteins were just extruded with no activity, but they were functional.
Okay, I see. Okay, though I think I guess that it is still possible that basically nets kind of damages most of the outer bacteria part, and then particularly maybe is harmful. I mean, it is interesting to see it, but I don't know if one can say just target specifically virulence factors. But thanks.
Yeah, about the specificity, that's true. We don't know if it's that specific.
Do you have any idea if these two virulence factors are secreted because OMP1 is in the membrane, so that's somehow could relate with all the things you have said, but I don't know if it's the fact that these proteins are soluble and that's why they are targeted by the nets. I don't know if you have any idea of that.
I'm not sure. I think one of them can have a soluble form, but I'm not completely sure. I would be guessing if I say so, but that's true. That's something to consider.
Okay, thanks. No, that's a good point about the secretion and instead the membrane tethered components. Because though, I mean, there are like some virulence factor, like I don't know, the type 3 system that are membrane also have got a part of the virulence there is a part of the virulence factor that is also membrane tethered. But I think in this point, maybe at this case, maybe that was the point I wanted to make. So basically, the pathogens are damaged by this, and they cannot even secrete the virulence factors, or the virulence factors are in a way also destroyed. So that's a good point.
Yeah, so someone raised a hand, please.
I just gonna add that elastase is specific. It has some kind of a specificity for aliphatic amino acids. I think it is related to the preference to degrade these virulence factors instead of another kind of proteins. I think it is related with the structure of the proteins it can degrade.
Okay, thank you.
Thanks.
Okay.
Carlos, do we have another person that wants to participate? Marijos Blanco, do you want to share us something?
Yeah. Hello,
Hi.
can you hear me?
Yeah.
Hi. Yeah, I have a question. It caught my attention that incubation with DNA intercalating dyes prevents the formation of nets. And I wanted to ask you if you know, like, what's the reason for that? Does it affects the charge of DNA or something, and that is why nets are not formed, or is it preventing signaling somehow? Hello?
Hey. I'm not sure if it's what I remember it said in the paper was that the DNA was labeled with the DNA intercalating. I didn't notice it was it prevented the net formation.
Yeah, they say textually that incubating with intercalating types of DNA prevents net formation. They don't show specifically why, or they don't apport sufficient information about it, but there is an interesting observation. I don't know why, what can be the mechanism.
So, hi. I was also thinking about that. It caught my attention when I was reading this paper, and I was thinking what could be possible reason for that. And I thought that perhaps when the dye intercalates with the DNA, it somehow makes its structure more rigid, and therefore the cell is not able to release it outside, like it cannot go through pores or something. Like, I don't know if that could be the reason, but you may have an opinion about that.
Yeah.
Yeah, also, as Dominica pointed out, I think it's important because this is something that also this lab published like 5 years later, is that some components of the primary granules translocate into the nuclei, and these components will generate different modifications in histones that will eventually relax the DNA, the chromatin, and this will allow the net formation. So this actually goes in line of what Dominica mentioned, that maybe the fact that intercalating dyes intercalate to DNA, this would not facilitate the process of chromatin decondensation.
Yeah, thank you very much.
So if there are no more questions on this part, we can continue.
Okay, so yeah. So then the other point that they have to make is, so until this point, they show that nets trap bacteria, no matter if it's positive or negative, or gram-positive or negative. They also show that this response is very specific to virulence factors. But now the point that they want to make is, like, what's the role of chromatin itself in this activity? Because at the beginning they say that they found primary granule components, but what's exactly the chromatin doing in this reaction? So one of the experiments that they do is that they evaluate the effect of nets in bacteria killing Shigella and also Staphylococcus, and they assess this effect by treating the nets with DNA. So, and they noticed that once they administer the DNA, the percentage of killing is significantly reduced. Additionally, if they use an antibody against histones, they have the same effect. And this is what lets them hypothesize that if you, basically, if you, that the structure of these nets and the structure specifically of the chromatin itself of these nets is the one that is exerting this antimicrobial effect. And in this video that they did, it's actually not very clear, but in this video, they have nets, and once they treat them with DNA, basically all the interactions are completely removed. So yeah, so in here the major claim is that it's not just the fact that primary granule proteins are trapped or an antimicrobial peptides are trapped in chromatin. It's not just the fact that this is happening. It's also there's also some effect in the structure of chromatin itself, and also there's an effect in the histones itself, promoting an antimicrobial effect.
Can I have a question?
Something? Yeah.
So I was thinking, because to me it looks a little bit like the histones and the DNA, which is wrapped around the histone. It makes a structure, so it's like a scaffold that traps those bacteria, and then the elastase and whatever other enzymes are involved can act to kill the bacteria. And without the scaffold, simply the bacteria doesn't stay in place for long enough, and the enzymes are also not in contact with bacteria. This doesn't happen, and this is the reason why without DNA or without histones, bacteria are not killed. So, but is there any other more sophisticated reason why the DNA and histones are so important for this process? Or is it just a mechanical scaffold that provides the proximity of enzymes and bacteria?
I think they in the paper also, and I think also previous reports show that, and it's kind of crazy, but if you do extracts of H2, I think they did it from some of H2 variants. If you do extracts of this histone, the histone itself, it have a antimicrobial activity, and this antimicrobial activity is even stronger than known antimicrobial peptides, the histone itself.
Yeah, but here in this mechanism, if you take one of those two, either a histone or a DNA, out of the equation, this means that this DNA scaffold is not formed, bacteria cannot be trapped, and therefore the histone cannot exert its antimicrobial activity towards bacteria. And same as the elastase, it's simply because they are not in close proximity anymore, because everything, it's dispersed.
Yeah.
Can I answer that?
Yeah, but what?
Try to.
Sure.
Yeah. So as long as I understand, what happens is that the DNA actually serves as an anchor and also makes the net structure a little bit sticky. And once the DNA serves as an anchor, exactly like something that just serves to maintain attached all the proteins and the histones. Histones themselves can act as with an antimicrobial activity. So I think there are different mechanisms. Yeah, it's not the same to taking out the DNA than to taking out the histones.
Okay, so then it means that the histone itself is responsible for bacterial killing. Elastase is responsible for like degrading the virulence factors, without which bacteria are still able to survive, right? This doesn't kill them.
Yeah. No, well, actually, yeah. The elastase, I think, serves to, like I said, like a sticky to maintain the bacteria attached, something like that, to trap them, and also to inhibit the virulence factors.
Okay.
But it's interesting, right? Because it would be interesting to have an experiment to kind of assess the contribution of histones, the contribution of nets as a trap, and the contribution of the elastase as antimicrobial factor, what's the word?
Yeah,
antivirulence factor.
It will be interesting to know how do histones kill bacteria. Yeah, because at least with this paper only, we cannot understand how that happens. And I think that's an interesting question. Thank you.
Someone else knows something about this?
Yes, look, Guadalupe.
Some of the histones interfere with the membrane of the pathogens, and other have another kind of mechanisms in order to kill pathogens. Some of them penetrate and interfere directly with the DNA of the pathogen, but some of the stones interfere with the membrane of the bacteria.
Yeah. Yeah. Okay, so
if there are no more comments, we please turn off your microphones, and we will continue with the paper discussion.
So the last figure of the paper is basically because until this moment, all these experiments were carried out in non-fully, like it was not in vivo conditions as this of what they refer to that as in vivo. And so they tested if the net formation also happens in vivo in models of shigellosis in rabbits and also in spontaneous appendicitis. And at least one of the things that they notice once they look at it in the tissue is that it also happens. And again, they have a co-localization of histone DNA close to Shigella specific LPS signal. And additionally, they notice that nets are.
Yeah, can I interrupt a bit? Because
yeah,
sorry, I just think that it would be important to mention before going to the in vivo experiments, to taking in account that in the in vitro experiments, it is important the time at which the nets are developed, and that time is different from apoptosis. So I think that is special to mention that. I think that in this paper, of course, it was the first one to describe netosis, and it was not clearly known the fact that there is vital netosis and lethal netosis. So there are two types of nets, the ones where the cell dies and the ones where the cell survives. And that is very interesting to compare the fact that I think they had a video or something like that in the supplementary data where they demonstrate that the time lapse of apoptosis is different from the time lapse of netosis. Just to keep all the stuff about the in vitro experiments before going on into the in vivo ones.
Yeah, I think that was very important.
Yeah, sorry, netosis, the release of nets occurs before apoptosis starts actually, but it doesn't mean that netosis is occurring. It because in this paper netosis is now the main mechanism they describe, because to cell start netosis, that involves so many steps before the net release. So I guess in this paper specifically, it's just the very first time that they observe the net release in the extracellular environment. So I don't know this if netosis could be involved in. Obviously, it's involved. Now we know that, but in this point, I guess it's not. They don't involve netosis as a mechanisms. Just they in the supplementary data they show that net release occurs before apoptosis start. Yes.
Thank you everyone for the comments. Does anyone have something else to add? Okay, then we can.
Oh, sorry.
Yep.
Sorry.
Yeah, go ahead.
I just found interesting that in a part in the text, I think it was like close to, here, close to the end of the paper, then at least for me, it was historically interesting to see that they already knew that neutrophils aged, even though they didn't probably knew how to define it. And it was interesting for me, like, after the discussion we had with Andres, that like in their 2018 paper, they defined molecularly what does it mean to that neutrophil was aging. But I was surprised that they already, at least they already managed to observe a difference in neutrophil shape, and they call it aging in vitro. I think that was like interesting, at least.
Yeah, and also I think that the fact that they asked the question in the paper about, we know that neutrophils die in vitro in a way that is like very soon, as the moment you put neutrophils into culture, they can die immediately. Yeah, they die so easily. But also that it's unknown the mechanism by which the neutrophils die in vivo. So they propose, as long as I understood, they propose that the mechanism of neutrophil death in vivo is related with nets, with net formation, at least.
True. Whereas now we know that netosis, for example, is very pro-inflammatory, and instead neutrophils normally die in a very anti-inflammatory way, and the macrophages are also involved in this. But it's interesting to historically understand then how people build up hypotheses.
Yeah.
That's a very nice insight. Thank you so much for sharing. So we can continue.
Yes. So in the last figure, as I mentioned a bit, they tested the function of they tested where the net formation would also occur in vivo, and they see a very similar phenotype in which they see these structures being formed, and also that the structures co-localize with the Shigella specific LPS. Additionally, they also claim that these neutrophils, these nets will aggregate in tissues, and that the nets will aggregate in clusters of cells. And I think this is important because this is what leads them to I guess, to put the other figure, is that in a spontaneous appendicitis in humans, they also see net formation, in which they see again the complexes and the colocalization of both DNA histones and the neutrophil elastases, and again they see these in clusters of cells. And this is what leads them to hypothesize that probably nets could be involved in autoimmune diseases, because the fact that they can be produced, especially in inflammatory sites, could result in an indirect toxic effect for the cell, which is, as Federico mentioned it from one of our previous journal clubs with Andres Hidalgo, this was exactly the main point of the study, like what happens with toxicity of neutrophils in steady state, basically, even if there's not an infection. So yeah, if someone wants to add something about this.
I think it's important to note that not all the neutrophils will undergo net formation. Like there are some that will do it, and some others may also kill bacteria using phagocytosis or other degranulation. So I think it's important to just keep in mind that not all the neutrophils will undergo netosis in a coordinated manner.
But I don't know if all the taking out your last point, if all neutrophils have the potential to do nets, or just like specifically some of them could. Do you have any idea?
No, I don't really know. I think
No.
it depends. Sorry.
No, go on. Sorry.
Yeah, no, I was just saying, as I wanted to remember the last discussion with Andres Hidalgo, that depending on the age of the neutrophil, the function was going to change. That was my comment. So that might affect net formation. But that's very interesting.
But also the way I understood it, I mean, reading from one of the Andres review, and from what I've read, I mean, it seems that yeah, neutrophil seems really to be like kind of programmed to do certain things and to react in a certain manner as soon as they leave the bone marrow. So like I think that it has never been established whether there are neutrophils that cannot or can do nets. So I think that's an interesting point. But I will like be for the party of, they can all do nets. But I think it's interesting also to think that we don't actually know now why neutrophils sometimes react by building up nets, and why they react by like doing phagocytosis, for example. I think it's a mixture between signaling, like between I think there is somehow a polarization involved here in terms of integrins and also pathogen recognition receptor signaling. Because I think, like I've seen it, for example, I read that neutrophils tend to react by netosis when they fight helminths. And it makes sense because helminths are huge, you can't phagocytose them. But I think that they maybe react by nets when, like, it doesn't make sense if you think about it, that they all react by nets, because then you need some neutrophils that start eating up those bacteria. So I think it's still a very fertile, field of research. That's what I want to say. Sorry.
Yeah, Angel Garcia wants to participate. Angel?
Yeah, another thing that we could say is that the neutrophils, they are polymorphonuclear cells. So in this case, we can say that its nucleus is, it could be bilobulated or trilobulated. So if we can remember that the net formation is not only by the DNA influence, we also consider that it could be influenced by this formation of the nucleus. So if we consider this on advanced age, it could be more the formation of the nets. Can we consider that point?
Yeah, I think that's true. I think also maybe that's a way to limit also the damage to DNA provoked by the granules, the thing of being lobated, but also, who knows, maybe a sort of way to delay the death by nets due to the release of DNA. I mean, but just an hypothesis, like I didn't read about it.
One of the earliest steps of net formation is the lobulation of the nucleus. So I think that maybe there is no principal role of the number of lobules of nuclei. So I don't know.
I think it's also interesting to know how the neutrophils decide which mechanism to use. I mean, as long as I understand, yes, all of them have the potential to do netosis and to do phagocytosis, both of them. But I don't know how the regulation works in this field. how does a neutrophil decide if it's time to explode, forming a net, or it's time to phagocyte or to produce chemokines or cytokines to induce an inflammatory environment? I don't know. So I think it's very interesting to know how these regulation networks work. I think they might be some epigenetic regulation, or I don't know, because all bacteria have, or at least all gram-negative bacteria have LPS, and not all of them are trapped in nets. Some of them are phagocytes. So I think that's very interesting. I don't know what do you think about it?
Yeah, but that is not only for bacteria killing, because the main action of nets is against fungal cells. So in this order, if you deplete neutrophils, you can get a defense against yeast or I5 of Candida albicans. So I will say that the way they can distinguish between different pathogens could be the cytosins involved in the medium, and then they can to choose to release nets or start netosis. So that also is important to say, as someone mentioned before, that release nets not implies to neutrophil death, because some neutrophils can live long before after release of nets.
Okay, Paulina Lopez Carrasco, do you want to participate after Jesus Benito, and then Marijos Blanco, then Rafael Navarro?
Hi, as Aranza said, I think that the media composed by interleucins can be responsible for changes at, I don't know, genome level that make some histones structurally different, and that could lead to a subpopulation of macrophages, neutrophils that can make netosis and the others that can make phagocytosis. Do you think that it could be that the interleucins that are in the media could be the responsible for helping the neutrophil decide if they want to make phagocytosis or they want to make netosis? Could that be a possibility, that the genome arrangement is more favorable to release the DNA or the nucleosomes than other type of arrangements that are not that are developed after certain other type of interleucins? So that could be the difference that could be caused by different bacteria.
I think at least by now, what is known is that the facts that can trigger the initiation of netosis are not necessarily cytokines. As long as I understand, I know some people that works in nets, and they use mainly stuff like PMA or LPS. There are no cytokines to induce netosis. So I think that cytokines cannot induce netosis in that easily way. I think the easiest way is just the fact of the activation of nets. There's spontaneous netosis actually. If you take neutrophils out of the blood and you purify them and you culture them, some of them will spontaneously explode and form a net. But I don't think that cytokines can be the main, like the main agent that can start netosis. I don't know, what do you think?
Yeah, for sure.
Like
the microenvironment is important, but there has to be like some signal of bacterial damage or infection to start the netosis. Well, of inflammation, let's say, because we can have also netosis with no bacteria infection.
Yeah, and actually,
So
sterile netosis participates in some diseases like lupus. I'm sorry, someone follows. Like,
yeah, if Jesus Benito, if you want to participate.
Yes, thank you. Do you hear me?
Yep.
I understand that the way that neutrophils decide between netosis and phagocytosis is related with the composition of the pathogen. For example, pathogens that can't be phagocytized because of the size or because of the presence of virulence factors trigger netosis, like for example, like it was mentioned before, some fungi that are too big to be phagocytized, they trigger netosis in neutrophils. And in the other side, little cells like bacteria are easier to be phagocytized than to trigger netosis.
Yeah, that's true. Have been some publications talking about how neutrophils can somehow sense the size of a pathogen and respond like accordingly. Thank you for that comment. Marijos Blanco.
Yeah, I have a related question. On the paper, they use PMA, a certain concentration of PMA to induce netosis. And I wanted to ask you guys if you know if this certain concentration, which I think is 25 nanomolars, is like, could be found biologically or physiologically. I don't know if it's like too high or too low.
I don't know. I think it's a very low concentration, but I cannot. I don't know.
I think it is very artificial. I think
Yeah.
that you cannot find PMA like naturally in the body of.
Yeah, it's actually a treatment.
PMA for sure you cannot find, but talking about the the treatment with IL-8.
Yeah.
An LPS, that's for sure something you can find in the body. But yeah, like PMA, it's like chemical compound. So yeah, it's just widely used to activate cells.
Okay, thank you. I had another question. Can I do it?
Yeah, sure.
At some point during the presentation, someone mentioned that nets do not necessarily kill neutrophils. And I don't know, I just wanted to ask, how could a neutrophil survive once a net is released? And whether, bringing back this point of what sign or how a neutrophil decides whether to phagocyte or to release a net, in this same sense, how a neutrophil decide to release a net and basically explode, or to release a net and then survive. I don't know if I made my point clear.
Yeah, both. I think it's still. Yeah, if someone wants to comment on that.
Yeah, I was the one who said that there is vital netosis and lethal netosis, and I think it's still not well understood how it happens. I think that there is some different regulation on interferon genes, but that's all I know. Yeah, I don't know how they decide to do one or another, and I think that it's a very unknown field in the area of net study. Yeah, I think that we need still a lot of research related to that.
But also I think that it firstly comes from the, for example, observation of like intravital microscopy. So in the space of in the time of a video of like 20 minutes video, maybe people would observe that those neutrophils that flesh out the nets then still are motile and are still like ongoing or like interacting with bacteria. But who knows, maybe in an hour they will be dead, for example. And I guess that, as I think Carlos said, it's very tough to then distinguish. Like, I think you will need somehow a technique of maybe of one of those molecular tagging, that you tag the neutrophil and then you see like his neutrophil tag, and like the one that doesn't die, and then you do a single cell RNA on it. But it's very, I mean, it's very expensive for something that, I mean, in the end, do we care? They are gonna die anyway. But but I think it's, yeah, I think we don't know the answer clearly.
Thank you very much. It's quite interesting.
So Raful Navarro, if you want to comment something real quick before we go with our questions and answers.
Hello.
Yep.
Okay, so regarding the last questions, I think my question will be if there is any difference between the stimuli involving net formation and the stimuli involving just a simple degranulation, or if it's just the strength of the stimuli that differences between these two types of responses.
I think there are differences, like net formation not always happens, and yeah, for sure the stimulus is the kind of stimulus that triggers it is very important. So thank you for the discussion. Carlos, if you want to finish with a summary of the paper before we go with the questions and answers. Okay. So
I think we better continue because, yeah, we have time.
Okay, so
we have to continue.
Today we are delighted to have Professor Arturo Ciklinski with us. So Professor Ciklinski studied biology at the Instituto Politécnico Nacional in Mexico City from 1980 to 1985. After that, he was accepted at the Rockefeller University in New York, where he received his PhD in immunology in 1991. After that, he spent 2 years as a postdoc in the Institut Pasteur in Paris. And when he finished this postdoctoral fellowship, he worked as a professor at the Skirball Institute in the Department of Microbiology at the New York University School of Medicine from 1983 to 2001, after which he was appointed director of the Department of Cellular Microbiology at the Max Planck Institute for Infection Biology. According to his website, he has 91 publications up to 2019, but we know that despite this pandemic, that number has increased in this year, 2020, and I'm sure it will keep growing. So welcome, Professor, and thank you for being with us today.
Thank you very much.
Yep. So the way we are going to do the questions is, please, if you can write in the chat a number. For example, if you want to ask, just write 1, and then the second will write 2 and 3, and then we can give you the, you can speak. And if you are also, you can also write the question in the chat.
Okay, I open the chat.
Yep. So yeah, we have the first question from Jose Pablo.
Yeah, hi, Professor. It's very nice to meet you. And I think that a question that we all have when we see papers related to nets is how do a neutrophil decide whether to do netosis or to keep doing phagocytosis or performing them, you know, cytokine production or stuff? I mean, how does a neutrophil decide when to do netosis? And also, how does it decide how to do vital netosis or lethal netosis? Thank you.
That's a very good question. It depends on the stimulus. So how to do, I cannot speak about vital netosis, we've never seen it, but there is a very interesting paper in Nature Immunology by Venizelos Papagiannopoulos. Sorry?
Sorry.
There is a very good paper by Venizelos Papagiannopoulos in Nature Immunology a few years back, where he showed that the neutrophil somehow measures the size of the stimulus. And the bigger the size of the stimulus, the more likely the neutrophil is to respond by making nets. So if you present a neutrophil, for example, with a high phi of a fungus, it always makes nets. If you present it with something much smaller, it then has to depend on the stimulus of the nets. We have worked very little in actually on the receptors that induce net formation, but I suspect that you have to engage several receptors in order to undergo netosis, right? It's a very final decision, and I don't know if you reviewed that before, but nets can be very dangerous, or nets are very dangerous if they are done at the wrong place and the wrong time. So it is an important decision, and I think it has to be integrate information from several receptors. So if you stimulate a single TOLF, if you add LPS to a neutrophil, or if you add only BLPs to a neutrophil, it's most likely to make cytokines. You really need a more complex stimulus in order to induce nets. Does that answer your question?
Yeah, thank you very much. And in this regard, I mean, small things like bacteria, like for example, Mycobacterium tuberculosis, can induce net formation. But
yes, there are papers on that.
Yeah.
I don't remember who wrote them. There is a Mexican group that reported that, but
yeah, it's the group of the Escuela Nacional of Ciencias Biológicas.
Yes, true.
Yeah,
But there are other reports that has been reproduced, right? But of course, we don't know if they are seeing a single bacterium or a clump of bacterium.
Okay.
That is very difficult to understand.
Yeah, thank you very much.
So the second question, Mosopani in the chat asks, is there a metabolic reprogramming before net formation, or something known about it? Thank you.
Another good question. We know very little about it. I know that there is a story that we have not published in my lab looking at patients that have a mutation in TALDO, which is an enzyme that is part of the pentose phosphate pathway, and they cannot make nets, but the data are not easy to interpret because in the pentose phosphate pathway, for example, is also required for the production of NADPH. And NADPH is one of the cofactors required for the NADPH oxidase, like the name indicates. So we don't know if they cannot make nets because they don't produce enough reactive oxygen species, or is there a real metabolic requirement for it? I think very few people have really looked at that question, but it's really interesting.
Thank you. So Gina Garcia Romo wants to ask the third question.
Yes, I don't know if you can hear me. Good morning. Okay, thank you. It's a pleasure to meet you, Arturo Szylinski. I was working with Virginia Pascual, and then in 2011, we published this paper with...
Of course, that's why your name sounds so familiar. Of course, you are the first author on the lupus paper, right?
Exactly, that's right. Well, my question is about the isolation of